mining on large scale, whatever the data parameter you're looking at, you need to think about what is the variability and how it was reported. Even getting down to taxonomic names, if they've changed over time, those old records may not be linked to the new name. So again, that can be kind of a gotcha when you're trying to do these kinds of analyses. Not sure what we're doing, but we'll get there. Okay, we're back. Great. So again, it's just some a food for thought. And then it was unfortunate, but we also saw almost 3,000 of these SRI records with no disease recorded. It's COVID-19. We did know the disease, but again, it wasn't filled in. So for data miners, looking at that data, it's definitely a concern. Now I want to look at geographic location. This is something we all use in many of our studies. How are we looking at this data? So again, for that same paper, we looked at geographic location in the SRA, and at the top of the slide is the data the analysis we did 2 years ago, and I repeated it this last week. To show you again, data is still an issue. One of my major concerns is when you get a geographic location filled in and it's empty or it's uncalculated. Don't know what uncalculated means, honestly. There's not a control vocabulary for that. So whatever that means, the data is missing. How do I do analysis on location of where variants are occurring and growing and evolving if the data is not reported? So again, this is just something food for thought. If you're pulling these data sets, look at the variability, understand where it is before you do your analysis, and then you can incorporate that in what you're looking at. So unfortunately, this is still an issue, ongoing issue, but again, I'm trying to encourage people to do better reporting and then kind of have some additional QC on what they're reporting. I'm not sure why at this point there's 3 different types of fields for geographic location. I suspect it's based on submitters and the fields that they're submitting, and they're under different standards. And that's okay, as long as you know to look at the 3, incorporate them all. You're good to go. Now I'm going to look a little bit about variation in metadata reporting. So these are randomly chosen records from the biosample repository. I'm not picking any particular study, but I wanted to give you representative examples of little things that I think in doing data mining at the scale you're doing are certainly gotchas. So osteosarcoma, okay, we all understand what that is. But when a field is filled in human osteosarcoma and you're data matching, will it match? That's something to consider. On the far side, you'll notice there's a space missing in the host name. So will your algorithm know that Homo sapiens without the space is the same with the space? That's, again, just kind of a gotcha. But it's something to think about. And the other field that I think gets reported in so many different ways is age. And I just pulled out some different examples in the box on the right. Whether it's a year, months, days, question mark, I'm not sure how you're going to interpret that, but you want to exclude it, right? So when you're doing the analysis, it's things to think about. Sometimes you're going to get phrases in there as well, so not just a value. So and I think it is always a part of this kind of data analysis that we do is to think about the cleanup, but to think about the fields as well. And one other that I didn't point out on the slide, but I've certainly seen a lot over the years, geographic location as lat long. Ensure it's the place that's actually listed as a geographic location, a physical location, because that can easily and has been variable. I certainly have reviewed papers out there where they have geographic location, X, Y, but it's not in the country that it was supposed to be. So anyway, something to think about. I'm showing this genome data viewer view of NCBI. It's a very simplified upper level taxonomy of the various eukaryotic genomes that have been sequenced, right? This is, again, very simplified, as I was saying, but it makes us think of what are we sequencing, to what depth, what variation. It's a perfectly good image, but again, makes you think about what's been done, what could be done in the future, and looking at the variability. Looking at big data, we often will take the bolus of data we're looking at and, do our analysis. But I always like to dig a little bit deeper into that data and see what's represented. So the first genome actually I worked on was a human genome project. That's where I worked at NCBI to start with, and then the various model organisms. So we do still have one representative assembly at all times. Fantastic. But what's under that assembly? What are the other genomes that are there? So I use human as an example. So I looked at the 1,000 or so human genomes that currently exist in NCBI's Genome Viewer, and then just kind of scrolled down the page and picked one at random to see what was represented. I wanted to see what was available as a data miner to look at. So what I'm showing you here is, you can see the various attributes that they're represented. So they had don't have age, but they do have the gender or the sex of the individual, and you will get both gender and sex under that category at times. It just kind of gets mishmashed. Anyway, what I really wanted to point out is there's no geographic location for this individual. Even a country, but there is a submitter lab. So the biomaterial provider, we know where the sample came from. So I was curious. I, Googled, and it turns out, it's from Taiwan. It doesn't mean the sample came from Taiwan, but the submitter was from Taiwan. So at least it's a little bit of information that you can mine. And so reason I put this up is I want you to think about when you're gathering these data sets, there is going to be more data even in the information you're given. But if you look in the publications, in the supplementary files, there's a lot of other information you can mine beyond just the initial data submission. Now I'm going to talk about FAIR challenges a lot. We hear about FAIR. I live in the NIH world of funding, and we talk about FAIR data quite a lot. But what I want to do here is talk about a challenge to all of you. When you're looking at your data, when you're depositing it, when you're accessing it, how is it fair? How can we make our data more standardized, more structured? How can we take that uncharacterized, unstructured data and turn it into something that's useful for further analysis? How can we create data sets that will allow that further analysis? So think about that as we go forward. Now, on the genomic data side, we do think about FAIR. We recently had a meeting, our annual meeting, first one since COVID, so that was quite nice. But we had our annual meeting, and we were talking about FAIR as well. And we're saying, wow, we were cool before FAIR was cool, which is actually made us all chuckle a little bit. But what it meant is that we were thinking about how do we make our data useful to the rest of the community. So the Genomic Standards Consortium, we got together in 2005, and I'll give you more history on a further slide, but we realized this pure tidal wave of data was coming in because we were all genomic researchers, sampling all sorts of things in the world, and realizing we had to have a way to get that out to the rest of the world beyond just our own study. So thinking about FAIR in the context of genomics, we need to have metadata that's standardized. We need to have identifiers that are persistent. We need to have languages that you can do analyses on. And these have to evolve with time. Excel files, grand. That's how we've done them everyone's done things for years, but it's obviously not cutting it. So there are many file types now we all use that we can exchange information. And again, we have to think about licensing. How are we allowing our data to be shared? Are we putting an explicit license on it? Are we putting it in an accessible database? I can't tell you how many times as an editor of the microbiome journal that I've had submissions and go, oh, I put that data in my own repository. It's accessible, no problem. Here's the link. And I'm like, nah, because the data policies are to make it fair that it has to be accessible to the public. No login, no restrictions, all of that. So again, we have to continue to think about where are we putting our data? How are we making it accessible to others? To again, always trying to have it more reusable. And really, in the end, we want our data reused, right? You want people to be able to do analysis. You want that to be part of the building of the bricks we all do, adding data and information to build that wall of information. We want that as our outcome of our studies. It's not just our graduate work being done or our postdoctoral work. We want our data to live in perpetuity as best as we can. But again, making it as fair as we absolutely can. Now, addressing biases in our data, I thought, again, my thoughts turned to CAMDA and what we were doing here. And no, I didn't do any of the CAMDA analysis, but I did look into a bit into what the data sets are. And so these are potential biases. It could be in any of the three areas that were done. But again, just making you think of what the context of the data you're analyzing is. What could be in there that could be maybe new areas of insight into it. Maybe there's variation in the geographic location of the AMR samples that you're doing. Maybe there's areas that were covered in the world and areas that weren't. So not just looking at what's in the data set, but what's not in the data set. What's the context? So that's what I wanted to really touch on here. Thinking about other things that could be involved that would impact the analysis you're doing and the outcomes of what you're looking at. So even the disease maps, modeling COVID-19, and it was brought up in the keynote this morning. We have a genetic variability we're looking at for all sorts of diseases. But again, we're looking at host genetics, we're looking at environmental determinants. There's a lot of other factors out there that could be impacting what we're looking at. When we're looking at genomics, we are also often having discussions in rooms just like this and saying, what don't we know? What haven't we discovered yet? So we mentioned the Metasub Consortium at the beginning of the talk. In that group, we often talk about, well, what haven't we discovered yet? What is that? You know, when you do your sequencing and you get all your results back, that nice pie chart of, we know, say 60% of the organisms that we sampled in the subway. But what is that 40 other percent that's the unknown? What's the, when you look at GenBank and there are function unknown sequences, what is that, or others call it microbial dark matter? What are things we just don't know? So I'm always questioning what don't we know? So here I'm just showing you a little bit of reporting from NCBI's genome counts. Again, thinking about, and that whole context of the tree of life for microbial genomics, where have we sequenced? Where haven't we sequenced? Where could we do it deeper? And this is just to give you some numbers. I mean, we all love numbers. We like to, you know, we see where our data is. So even bacteriophages, only 392 genome records for those. So where are their data? Where is their data rich? Where is their data that has opportunities for new studies? So that's why I put this up, to really just think about where is that diversity and where are there areas of opportunity for us to go further. Now, as part of a group, we spent about 2 years looking at this very question. So this is our publication that just came out this year. We looked at, and it did take us a while to figure out the title, but Darwinian genomics and the diversity in the tree of life, really looking at what has been done in the last 2 decades in our field. Where could we go? What are the questions we can ask with all sorts of other organisms? Again, thinking about what's the data that we have represented, what data haven't we represented, and really trying to get a perspective on where we've been spending all of our focus, and are there other questions out there we want to ask. So that's what the gist of this was, really just to bring out the numbers though, and looking at how many eukaryotic species have been sequenced and where do they have their representation in GenBank. I'm going to show you some data we pulled. There's two, I'll go on the left side of the slide first. So first, we pulled up the numbers. This is all from the SRA at the time. Looking at who was represented, whether you're pulling out just pure amounts of data in terabytes, or were you looking at individual records. And again, I mean, it wasn't necessarily an obvious conclusion, but again, if you don't have numbers, how do you talk about it, right? So we really wanted to understand where the diversity was, as we're thinking about, with this paper, proposing where we could be going in our next decade of work. And obviously, you know, as you can see here, human genomes have been sequenced predominantly over others, not in total, but you know, it's certainly higher. And this is for the last 10 years. No, yes, 10 years, sorry. 10 years of work. So again, on the right side, we looked at it slightly differently. We want to see at the diversity of species, how it played out, and what were the changing pieces of that pie I mentioned earlier, and how that went. And you can see that for other organisms, it's been decreasing over time. But the last one is really looking at species that aren't necessarily commercially or medically relevant. How much of those genomes have been sequenced. Again, I mean, obviously we had a point of view with this paper, wanting to have diversity in the organisms we're looking at, but also celebrating the amazing things that the various model organism databases have done, and the data of the genomics of non-human organisms, and where that's really enriching our knowledge base. Now, again, I wanted to think about potential areas of bias, and I'm bringing up a slightly different topic. This is my colleague at NIST, National Institute of Standards and Technology. So Scott leads a group that looks at this particular thing on the laboratory side of things, not the metadata necessarily, but the laboratory. And so we talk about this a lot. This is a slide he shared with me. And again, thinking about from that time we are sampling the wastewater, or the soil or the subway, from that very moment, what are the various biases or points of bias we could be bringing into the study? And the reason we've all been talking about this, and we talk about certainly in Metasub, is each of these points in time does add different biases in. Even in, and I'm sure Crystal will talk about it, the many countries are involved in Metasub. How do we standardize even the extractions? So we're using the same protocol. How about the sequencers? But then again, also the downstream bioinformatics pipeline. Think of how many pipelines we're all using to analyze different data sets, and where are the variabilities coming in our results based on any of these pieces. So again, in many of the projects I've been involved in, we look at all these pieces and then try and standardize the heck out of them, have as many points of unity across them, but also reporting. whether it's Unity or not, making sure you're reporting all these pieces. So someone then looking at your data set can then go, oh yeah, okay, I see where that you use this sequencer or that pipeline, and maybe I'll run the data on my new pipeline and see if I get a variability. But again, allows you to ask those questions. Talking about genetic disorders and thinking about our knowledge and how it's changed over time. So I know when I was in undergrad and grad school, we were thinking one gene, one disease, right? We got them, we'll figure all those out, we'll characterize them. But certainly in the last 20 years, all of our knowledge has changed quite a bit. We've grown, we've evolved. And the way, so I represent data diseases in our standard, but then that's how we all think about them as well, is it's changed and it's evolved to have all these different components. So sequence alterations, chromosomal abnormalities, looking at complex diseases in new ways, looking at sporadic mutations, somatic mutations. So the field of disease has changed dramatically since we started the disease ontology 2 decades ago. Again, when you're standardizing data, it's always informed, whether it was the genomic or the disease, informed by the science. Where's our world going? What discoveries are we doing? And how does that evolve? And then the standard has to represent that, because it's all built on the data, right? So, but it's something to think about, that when you're analyzing data over time, data may have been represented, 15 years ago, and maybe they were only thinking monogenic, and they hadn't really started thinking of digenic, or how one gene's impacting another, and that's impacting the outcome. So again, it makes you think about the variability in the data, but also I think it's also very promising of the growth and the ways we can look at the data. And just some examples here. I have a clinician team that works with me on the disease ontology, and we were pulling out our, I think we thought would be our most representative examples of this, this progression, environmental to genetic diseases. So some diseases are still considered completely genetic, and you'll see those at the top of the screen. So Tay-Sachs disease, we know the gene, grand. That's one of those, really good early examples. But then you get, you think ricin at the bottom of the screen. Okay, that's environmental, no problem. But just move up one, fetal alcohol syndrome. For the longest time, always considered purely environmental. It's alcohol, right? At a certain stage of development, alcohol exposure in utero. However, more recently, in the last number of years, a number of papers are saying that there's a variability in the maternal's host, in the mom, ways to ability in metabolizing that very alcohol. So that genetic variability of the mom, again, is playing a role. So as cut and dried as we all thought it might be at times, clearly majority of our work is in the middle. There's lots of middle, right? So even if you think of a purely genetic disease, there's often, there can be environmental context. And that's what we're looking at more, and trying to integrate that into our diagnostics, into our knowledge base, pulling that information, really trying to understand where we have definitively identified that, say, UV light in retinitis pigmentosa is a factor in the progression of the disease. So it's that second drop of the genetic, and then you got the environmental. So again, you see more and more of this. My team's put together a model that we are utilizing to look at all these complex diseases, really trying to get a handle on all the variability of what's out there beyond and the very much variability under the genetic side, pulling more of that in, looking at the epigenetics, looking at the copy number variants, as the last keynote was talking about. There's a lot of that information coming out now in literature, but also looking at social determinants of health, looking at other host factors. But as you know, there's always the unknown, top of the slide, right? We always realize that there's components of what we do that we're just not going to have an answer for, and then we're looking for those. We're looking for the diseases where we can't figure out all of it, and that gives us again a place to start to look at what don't we know. We've been spending a lot of time this last year looking at environmental drivers and really integrating that into our classification of human diseases, where we know the causal factor. There is a large body of literature as well where it's not known to be the causal factor yet, but there's hints. So we're keeping track of those as well, where there is a potential causal factor. But when we're classifying human disease, we want to only have the known factors. But again, this model is allowing us to pull those in, to assess them, and to give clinicians and researchers further tools for assessing their disease data. One of the outcomes of this work, as we go through the various complex diseases, is to reassess from literature and the clinician's team knowledge. We spend several months on each disease, and this is the outcome for the asthma that we updated this last year. Again, looking at the subtypes of disease beyond what was always classically known when we were teach in medical school. But again, thinking about other factors that are involved. So phenotypes obviously are part of this work, but then we now have endotypes. Endotypes in complex diseases, more looking at the pathophysiology components of the disease, and these are all published in published bits of information that are all incorporated into the ontology. But again, giving us a fuller view and giving our clinicians a fuller view when they have a patient that comes in with asthma, what are the possibilities of what that diagnosis could be. But for data miners, again, looking at more finite representations of these diseases that have these various components pulled together, so you can actually have hopefully a more informed review of the data and insights into it. I did talk about a few times the disease ontology, so I thought it was only fair to pull it up. This is our website. This is a project that's been going for about 20 years now. We pull in every type of disease, complex, rare, environmental, cancer. We have our latest paper on the NAR database issue this year. In January it came out. So if you're interested in seeing more details about that, but again, this is for data gathering. A lot of groups, over 1,500 now, cite us, but many of these papers are groups doing the data mining type work that CAMDA is doing. So looking in deeper into what the data is, pulling it out, looking at factors, trying to understand the diversity of data, the different factors involved. So this is our website. It's publicly available. If you have any questions, please let me know. There's all sorts of information on the website as well. Now, I want to look a little bit about rare diseases and the biases we may have when we're looking at them as a data set. So there's a lot of challenges in doing this. So if you look around the world and how the world classifies rare disease, it's close. I mean, it's close. The values are close, but they do vary. Country, and I listed them in the middle. They're based on either a law or a new review by a community. But these are the latest numbers of what they are. Even the number of rare diseases is variable. So if in the United States, the numbers you see most often is 7,000, an estimate. But if you go around the world, it's anywhere between 6,000 and 8,000. Again, people look at it slightly differently. And there's lots of different databases that include rare diseases, but there can be biases in the reporting. That's why I'm going to bring it up today. There could be biases in the interpretation. So rare in one place may be common somewhere else. And I do put cystic fibrosis as an example at the bottom. But there's many instances where a disease is common in one population or one community, one geographic location, but rare somewhere else. So again, it's something to consider when looking at data as rare. What these numbers mean and what the geographic aspects of it are. Okay. Again, I want to think about the genomic side and potential biases of things we look at, and ancestry is one of those factors that certainly comes, whether it's GWAS data. First, I was looking at dbGaP at in NIH, or looking at a GWAS catalog that NHGRI and EBI collaborated on. As you'll see from the figures, there's variation, right? And so there can be biases on where the data has been collected over time, and that's just can be quite honestly the reality of where their funding was or the types of studies that were being conducted. And there's nothing wrong with that. I'm not saying that. What I'm saying is it's good to consider what the variability of the data you're looking at could be. To look a little deeper into what would the analysis, and there's quite a large body of literature, and I didn't unfortunately have time within this presentation to go over it, but there's a large body of literature looking at like South America, Native Americans in the United States, what the biases in our data sets that we're analyzing, and where maybe we could put resources, or maybe where there are new studies being done to look at that variability that we don't currently have access to. So that's what I wanted to call out here, really just thinking about what is the data we're looking at, and maybe where we may want to expand that data. I'm going to give you a couple examples of approaches where groups I work with, collaborate with, have taken on this challenge of standardizing data that comes from not just many studies, but think about the diversity of the data in the literature that we pull from. So how many, if you're interested in a particular cancer, you may be going, go to PubMed, do your search and grab all sorts of data out, but realizing that that data can span decades, right? And the knowledge about that particular disease may change. The verbiage, or we also call the nomenclature of that disease, may change. So I'm going to give you some examples of groups and products I'm involved in where we're kind of tackling that very standardization effort. So what I'm showing you here is we had a presentation this April at the American Association for Cancer Research. And this is the research group that's involved. Jason Saliba, he leads this group. And what we do is we're looking at pediatric cancers in particular and the variation on how we're handling that data and how we can improve that data representation. This is really a collaboration between 4 groups. So yes, my disease ontology is involved, but also ClinGen, hosting a lot of this data, a huge number of researchers, clinicians involved in analyzing the data, Civic, clinical interpretations of variants in cancer, which is a great resource where people are reading papers, assessing those variants and annotating, I'll show you, but are annotating that data into a system that can be is regularized and can be queried. And then also looking at machine learning. So Jake Lever, he's in Glasgow University, again, doing looking at how we're standardizing our data for machine learning and having that machine learning data feed back to our system, looking at synonyms, alternative names, how we're representing the data so we again can do a better job. And again, he's looking at biomarkers in cancer and feeding that back into the system. So there's a lot of standardization aspects to one of these approaches. This I thought illustrated that very thing in this example. This is the Civic database. I just pulled out part of one of their pages. These are called evidence items. And what you're seeing here is how the various information is pulled out of literature by the curators. But what I'm showing you on the bottom is that this working group, we identified the age of onset as being a very critical factor. We couldn't, at that point, even just 2 years ago, query the variants, cancer variant data. So we went hunting. We looked for an ontology where it had that data already, like the age of onset data and defined in fields where it was done. And we did find most of it in the human phenotype ontology. They had the categories already defined. And so we adopted that ontology. But then as we did the curation, we realized that the young adult category was not refined enough for the data we need. The cancer variant impacts at these different ages really need to be defined in these other windows. So we worked with them and we were able to do that and integrate that. And now it's being used both in Civic and in ClinGen for our annotations. And I did put the approach, I listed out the bits there on the side of the slide for your interest. But again, this is a community effort, figuring out what the data problem is, figuring out a standard we could use, and then getting that integrated so it's in our system now and we can do those annotations. And again, what we're really doing at the end is we have now an open access, shareable data set of pediatric cancer variants based on age of onset. So again, that's where it's getting us to. One other aspect of this is, again, review of where the data is, current nomenclature. The WHO puts out new, call them books, every year, but not on every cancer. So they're in different groups of cancer. So the International Classification of Diseases for Oncology is one of their standards. So in this standards where all the new nomenclature comes out. So all the molecular subtypes of a particular cancer. So this working group, again, uses that standard to improve our interpretation and to bring in those terminologies into the disease ontology, allows us to do a better job of annotating the data, but also gets us to look at the perspective of some disease names are held in databases and they're older names and they're valid, but as you get new subtypes coming in, how do you align those two together to make sense of the data? You can't just throw out all the old data, right, when the nomenclature changes. You have to think about how it comes together. And this, again, gives us that opportunity to improve our representation. And we work through, in this group, working through different areas of cancers, like ependymomas we did a couple of years ago. We were just finishing up leukemias, and we're doing brain cancers next. We're bringing in clinicians from around the world who are experts in these fields, looking at the molecular subtypes, again, improving the data. So this is an approach of standardization, having a problem, and then using the standards to improve the data. And I just have 2 slides, and they're really brief. two papers where they're using a disease ontology, and that's not why I'm showing to you. The only reason I'm showing to you is I really liked how their figures represented the role of standards in these efforts. We talk a lot about machine learning and AI, and I think for me it helps, and I think it helps to have a little bit of clarification on what are the points in these systems where that standard comes into play. And so this is one example of just machine learning, and it's at a certain point for the training the data set. And there's the publication for you to look up if you're interested. And then just one AI one. And this one I love mostly just for the figure. They're using AI for cardiovascular events. They really wanted to understand, could they predict the incidence or reoccurrence of events in cardiovascular disease through AI. But the aspect of showing here is the figure really is quite clear in how they're pulling their patients out. They're looking at these particular traits, and the traits, these features, that's where the standards come into play. Whatever the features or traits are, that's where standards can actually improve and help the AI effort, because then you have the same set of terms you're using across the entire analysis, so you can actually have a hopefully more accurate picture of what's going on in the real world, then model it and look at the AI. So there's that paper for reference. Now, standardization of metadata, as I was saying, the Genomic Standards Consortium has been around for about 2 decades. We really had to think about this a lot and how it's important. And these are just showing you a number of projects that have grown out of these efforts over the years, where really it really exemplified how important it was to have standardized representation of all these different data types we were collecting as we were collecting samples and sequencing data, and really just showing how these efforts are ongoing. But again, to have big data analysis, you really do need those standards in the back end. Now, as we evolve standardization approaches over time, I find there are different types, and that's what this slide is supposed to show you. There are and they're all perfectly valid, wonderful for the situation they're in, but there's options of what you can be doing. So there are many projects where you develop a standard in-house, right? You have your study, you standardize your terms you're using, it works perfectly fine. But maybe you want to aggregate that data with other groups, and so maybe you'll adopt a particular standard, whether it's for cells or phenotypes or anatomy or diseases. And I put the Alliance of Genome Resources here because it's an example of what they've done. They have data and a number of modelers and databases, distinct systems, been going for decades, and then they want to align some of that data, and they're using standards to bring that gene information together. They bring the disease information, anatomy together. But then you have other sources where, and I put the example of the Common Fund Data Ecosystems project I've worked on. But this is a case where the Common Fund at NIH funded many different projects over the years, and then a number of years ago, maybe it was 5 or so years ago, the thought came to being that, well, maybe we want to be able to connect data through the systems. And you're not going to have all these different systems that have been built, funded differently. You're not going to bring them together. In this case, you're going to map between them. You're going to find key elements of data that have commonality, identify standards, you can map that data. And I think one of the wins of this, you're not asking the people that have built those systems to change. You're just creating a possibility of a linkage between them. And that connectivity of the data is the standardization role. And I'll talk about a couple other examples in the last set of slides. But again, we're thinking about different ways we can address the machine learning or AI challenges by doing some of these different things. And there's lots of different opportunities depending on what you need for your resource or your project. Now, I mentioned the Genomic Standards Consortium, so I'm going to put up our website for you. This actually just got revised, I guess, the way we've been going. How did I say, 20 years? So we put a new website out recently, so it's a new picture of it. But what we really wanted to show you here is we have different approaches for addressing genomic resource challenges. And so the GSE is an example of this. We have these things called checklists, and I'll show them to you. Excuse me. It allows you to capture all that metadata in a very rich way, in a modular way. So if you're all capturing, no matter what type of study you're doing or environment you're doing, you're capturing geographic location, collection date, host, all these different things are the same type of data, and we should be collecting them defined as the same way, using the same parameters. And this allows us to do this over time. So there's lots of information on the website. I wanted to put it in here, and it's for your information there. And we also have a GitHub repository. All the data is open and accessible, all those good things. But I want to show you again a timeline of development. So if you're building a standards effort, and many I've worked with actually follow kind of this progression. You're bringing a community together, just like the CAMDA community. You're finding common challenges And then you're developing solutions, but then you're growing by bringing more members of that other communities together. What are those challenges that are ongoing? The challenges we faced in 2005 are not the challenges we face today. Not the types of data. We're not looking at the same environments. We all started with soil and water. That was the genomes we looked at in 2005. And look at the diversity, the far right of the slide: agriculture, food, symbionts, viruses, metagenomes, and genomes. The world has changed, and then this is how kind of standards examples can evolve with it. I mentioned some of the standards earlier. These are just some of the publications. Again, this is a way for standards to share with the community, to get that communication out there that we have them. But again, this is just a diversity of different standards. And what I wanted to point out on this slide is when we all started in a room with maybe 20 of us in the room, we've grown to a community of several hundred. I think we're about 350, 400 individuals now in the consortium. From just standardizing the data we were collecting ourselves in our own sequencing centers, now this standard is being utilized by over 1.5 million, is annotated to 1.5 million records in biosample, and it's only growing at this point. But again, it's something it's built for the community, again, by the community. So again, a standardized approach for these different types of data, biosynthetic gene clusters, for example, all very different communities, but again, having a way to talk to each other, and that's the role of standards. And I wanted to show you a COVID related example. So this is Will Show. He's a colleague of mine in British Columbia. And we worked together for years on genomic and epidemiology resources. So when COVID came to be, one of his areas of his work went to is looking at how do we handle all the COVID data that's coming in. And this is a project that's now gone worldwide. But again, thinking of what are the standardization challenges, what are the tools and resources out there that I can reutilize, or I can pull in from colleagues, or that are available out there for me to use. And again, to face this challenge of what do I do with all this data. And this is some of the work they did and some of how they built their data standard, things that they were looking at. They were utilizing other standards that are out there, pulling ontologies that were pertinent for their data sets. And I think on the large scale projects, this is all what people have been doing. They look at what's available out there. What can I reuse? What can I go and tag and say, hey, can I use that? Can we alter it a little bit like this? We get, people talk to us like this all the time. How do we evolve it so it fits my needs, but then it's as usable to the community, as fair to the community as possible? And so these are the examples. And the last slide for that work, I want to really show you, this is their most recent paper. But again, the diversity of users around the world being able to use it. Again, part of doing any of the standards work is finding that community, getting them part of the conversation and building these standardized approaches, getting their voice in the room. So if all of us in this room had a different data set we want to analyze, and we need to get that standard in place, talking together, getting it part of the system, then it has the buy-in from that community, but you also have the voice of the community informing it. And that's really, I think, why the GSC and all these standards work have continued to evolve in the last 20 years, because we do continue to work with the community and pull new members in. And the last example I want to talk about is the NMDC, the National Microbiome Data Collaborative. This has come out of the Department of Energy and the Joint Genome Institute. Again, it's another approach where it's addressing a problem in the informatics side of all the standardization work we've been doing. Again, we didn't have a common infrastructure for a lot of the analyses. And if you weren't in a large sequencing center or a genomic kind of powerhouse, you maybe didn't have the resources to do some of these analyses. So it's democratizing that area of work. It also has a major aspect of outreach. So 4 of the national labs are the main funded group. Emily Elo for Josh, she's actually part of the Genomic Standards Consortium, but she runs this project for the DOE. As a PI, she's bringing together these different groups, but having this figure really does show you the different aspects of what they're trying to achieve. Getting the omics data and getting the metadata and having the analysis, but also having a way for people to standardize what they're doing across all of that. Here I'm showing you a figure of one of her slides where you do have the community outreach, but here I'm showing you the workflow. How do they devise? Where are all these data going to come from? Where are the analysis going to be? How do we have a shared infrastructure so that people can do analyses and take advantage of the tools that exist? So this is, I want to just make sure I pointed this out. I think it's actually a really interesting way to take on this challenge. And the NMDC has again been refunded. So this is the URL is there. Please check it out if you're interested in doing some analyses. They have all sorts of support for people. I think it's a really great effort. And I did include one last slide though on their workflow in case you want to know particular programs or omics tools that they have. They're on the site. It's quite diverse, from all sorts of omics. But again, to really give you, it's a very different type of, it's not just the standards, but thinking about all the components across the entire workflow, where all the pieces come together and make that possible. Okay. Now I'm getting just to the last slide. I wanted to ask the community here again to think about what can you do to address these fairness challenges in your own work, in future work, talking about data management plans, when you across the whole life cycle of your projects, when you're planning it, when you're collecting, when you're publishing, where can you share it? Where can you make it more AI ready, more fair? But again, thinking about this as part of your actual planning when you're doing the project, not years ago, data collection, people would finish the study and we get a file and say, oh, here's the metadata, please put it in the system. But it was always after the fact, right? And now the community has shifted to where it's part of the planning from the very beginning. And I think this is a great way to think about any of the data you're collecting and how we can do it better and how we can bring it together. So just a fun slide at the end. How AI ready are we? It's certainly something I've had to think about in my own systems. How am I bringing data availability to the fore for the AI community, and so it can actually be reused. And I would be remiss if I didn't show you my people. So here you go. This is my clinical team and my informatics team to build the human disease ontology. And then lastly, the Genomic Standards Consortium. These are our board members, all their various projects that have been involved over the years around the world to bring all the information together. And that's the end of my talk, and I thank you for your time.
Okay, it seems to work. Okay, thank you, Lynn, for your amazing presentation. I think if you have a question, please use the microphone, which is in the hall.
Hi, thank you for your. Is this on? Yeah. Thank you for your talk. I was wondering, you mentioned briefly about mining publications and supplements, and then reattaching metadata in those back to database accessions. I'm wondering, have you had any success with automating that process, and how does that scale to very large data sets, or is it just the human component?
Yeah. I personally haven't had haven't approached that to automate it. I've been involved in jamborees where we've done that. We've actually taken series of publications and had a group like this come together and pull that data out and then put that into a new database. And that's been very effective. I don't know if it could be automated. I'm sure it could be, but the data can be found in the legends of figures and in supplementary files. So that's a tricky problem to be able to, and I haven't seen anyone approaching that yet, but that would be a great project.
So maybe, more questions? David?
Thank you for that fantastic talk. I think it really shows the wide area of where still work needs to be done to make this all accessible, and particularly when we want to use automated methods like AI for inference. The old adage, garbage in, garbage out, applies nowhere more than here. And I was just wondering, because this is so much hard work, and I'm very impressed what you've showed here. I was just wondering, to what degree do you think one could also come from the other side rather than preparing it for AI? To what degree could one use AI to actually help with the cleanup? And as you mentioned, when things are in the legend and so on, I guess, or the different ways age are presented, or so these are non-trivial things. So I wouldn't expect AI to be able to automate this, from day one, but perhaps it could help with identifying possible misspellings, possible alternative representations of the same thing. Maybe it could find an underlying encoding in feature space that suggests that two different ways of saying something is actually the same thing. Have you at all explored that, or do you have any thoughts about this?
Yeah, that was a great question. And actually, very much so what you were saying, it very much answers it as well. So yes, I work with a number of groups who've done exactly that. So we've used either machine learning or AI to pull large vocabularies together and say, where are the differences? Where are the semantic differences, and where are the actual differences? And actually be able to pull in, identify new data connections for it. So yeah, AI and machine learning has been great great because you can do data cleanup. You can look at representations going up and down. An ontology is a hierarchy. So you can use that hierarchy information to actually identify new potential matches between different data sets. And I have different collaborators that are doing that.
Thank you so much.
Oh, thank you.
Hi, terrific talk. Thank you very
Thank you.
much. Two questions. First, if you're starting up and creating a new data store for genomic data, for example, and you're looking at the universe of ontologies, and the disease ontology is one that stands out, do you have any particular advice for where one starts? There's a constellation of ontologies and things
There
that
are.
one could use. And secondly, and a somewhat related question, we put a lot of in looking at the constellation of ontologies, a lot of work in describing the disease in finer and finer grain detail.
Yes.
Can you speak to anything? Are there any, to your knowledge, is there work going on in looking at ontologies of outcomes, meaning mapping it from the other side in terms of what's actually done with diseases and looking at sort of future states where we're trying to work backwards from patient benefit?
Thank you. Yeah. So if you have a new system and you want to build a standardized system, so the Common Fund data ecosystem is a great example of that. We did exactly that. So we looked at the world of different systems that existed, who and what were they using and what worked well and what had difficulties. And so we pulled the knowledge in from people, experts in the field, and figured out what was the best ones to. And then we started working with each of those ontology people to work it. The modelers and databases is a great example. Systems that have been running for a couple of decades, seeing how they have been handling the data, then informs the new system. And that's exactly what the Common Fund did to do it. For your second question, can you?
So looking at outcomes.
Outcomes, yes, okay. Yeah, so I don't know of a standard or an ontology that does the outcomes. We have, in the human disease ontology, kept it quite narrow into our scope, and then that's quite purposeful over 20 years, because as you can imagine, there's all sorts of things that get pulled in. Injuries is one of the more recent ones people are asking me to handle and to figure out how to do it. But outcomes, no, I don't know of another system that does that, but that's a great idea.
Thank you very much for a great talk on
Thank
this
you.
very important topic. I have a question regarding the incentives, actually, because from the incentives for scientists to provide standardized data sets for data sets. And since the beginning of this concept or understanding, there's also the ongoing discussion, okay, how can we incite, what incentives to the scientists can be provided? Yeah. And there were some ideas that it should be done by funding agencies, by publishers, but it didn't work very well. And do you have any, can you comment if it's better now, or there are any other ideas actually how to, let's say, enforce the scientific community? Because it's kind of boring part. You have to provide the metadata,
it can be,
the
yeah,
description of the data set and things like that.
Yeah, so question of incentives comes up a lot in our discussion. It's the whole carrot versus stick, right? And so I now, and actually we had a hand in this. Maybe 10 years ago, we had a meeting at NIH with the various funding agencies and asking them, how can we encourage the use of standards? And if you're putting in a grant at NIH these days, you have to specify what standard you're going to use. But incentive from coming from a genome sequencing center like I do, what is the incentive of someone to share their data, which there's always the concern someone could be reusing your data, right? Or in an aggregated analysis. But the incentive, I think, is more community oriented, and that you want to have a better data set for that community. There's always a tough thing. There is no stick out there currently. You are supposed to report it as part of your data, but I think it's more of a community push. We found when we had projects like Cheetah, Chime, MGRAS, JGI's, Gold Data, when the large scale producers of data are doing a certain standard, it pushes a bit of a pressure on other communities of best practices. I think CAMDA actually is again a great example of what are the best practices in the community. You're leading it, and then you're encouraging other individuals to then do it. So the incentive, I think, is seen in things like CAMDA. Certainly the MetaSub consortium, there was incentive to share because then we can have the analyses together. There is no unified, unfortunately, incentive system. People have been using ORCID IDs, and we've been using them more and more to recognize people's individual efforts and recording those in systems. And those as another carrot for people's CV and for their projects.
Thank you. I don't know, because we should maybe see the questions from the virtual participants.
Okay, and we're good.
What are we seeing? Somewhere?
Okay. Okay.
So I don't know whether this can be heard by the people that are on the virtual channel, but overall we have had over 150 people attending, and so far we had questions so far only from people on site. So if the virtual people hear this, please, both for this talk and future talks, please do engage through the app, because that is the only way we can see your questions. Thank you.
Can I just have one last comment? I think the other incentive is the journals. The journals have been pushing their policy, not pushing, they're sharing their policies. The community has been asking to have the standard, and that is a big incentive to get into these journals. And your data is much more represented. Your paper is going to be more cited. I think it's a better way to represent the information. So again, keep talking to the journals and try and publish in journals that have these nice policies. Yeah, scientific data, Giga Science is right there. Yeah, so it's a good woo-woo. But thank you. Yeah, I think the journals is one of our best ways to push our agenda forward.